Hello, dear students. Good morning to all. Today we will learn about one of the important and core topic in cell biology and molecular biology is DNA replication in prokaryotes. This topic included in 5th semester BSc final year botany subject. As per central dogma of biology, the three main important processes like replication, transcription, and translation are very important to maintain the genetic information by all cells. This genetic information encoded in DNA molecule transferred into various gene products like RNAs, proteins, enzymes, and depending upon other genes also. DNA replication is a fundamental and biological process. In this process, the genetic information transferred from one generation to another. In this complex process, a parent DNA molecule undergo division. It produces two identical daughter DNA molecules at every cell division process. These two daughter molecules are generated and transferred into two daughter cells. This process occurs at a synthesis phase or S phase in interphase of cell cycle. And this DNA molecule should be act as a main genetic material in most of the organisms. And moreover, DNA molecule also called as master chemical of the cell because it conserves the entire genome for the next generation. What are the three possible replication patterns in different organisms? The replication process done in three models. One is conservative model, second one is semi-conservative model, third one is dispersive model. In the conservative model, after completion of the replication process, two daughter DNA molecules are produced. In two daughter DNA molecules, one of the daughter DNA molecule contain two old strands, and in another one, two new strands. That means one complete DNA molecule as usual with parental type, and the second one is completely new one, that is called conservative DNA replication model. The second one is semi-conservative model. In this model, two daughter DNA molecules are produced. Among the two daughter DNA molecules, one of the strand is always old one or parental one. Second one is newly synthesized. In this way, 50% of the parental genetic information conserved at every generation. That's why it is called as semi-conservative model DNA replication. In the semi-conservative DNA model, you can see one strand is completely similar to the parental, and another blue color strand is newly synthesized. So each daughter DNA molecule contain one new strand, another one is old one. The third one is called dispersive DNA model replication. In this DNA replication process, the parental DNA molecule divided into small pieces, and each piece produce the respective new strand. These new and old strands are mixed up with each other, and it is reattached and form a complete molecule. So that's why we can see in the photograph a portion of the daughter DNA molecule with the parent one and another portion with newly synthesized. So totally mix up the all strands and the reunion takes place and it gives a complete molecule. These are the three types of DNA replication patterns. But semi-conservative semi-conservative replication model is proved by Michelson-Stahl experiment in 1958. There was demonstrated a experiment with a bacterial species. One of the bacterial cell grown in N15 nitrogen containing medium. So these bacterium cells undergo binary fission and replicate the DNA material and produced several generations. At certain stage, these bacteria cells are suddenly changed into culture media containing N14 isotope. Previously it is N15, that means the DNA polynucleotide chain containing N15 nitrogen bases. But later on, suddenly these bacteria cells are transferred into culture media containing N14 isotope. So after first generation formed during the growth of the bacterial colony, in the first generation we can see all are intermediate type of DNA molecules. Intermediate type of DNA molecules means first one of the strand with N15 isotope, opposite strand with N14 isotope. Heavy chain is there and light chain is there. So in the first generation. After the second generation, or in the second generation, after first generation, we can see four molecules are formed in second generation. In the second generation, two molecules with half of the high heavy chains and half of the light chains. So two completely with heavy chains. See here, these are this strand produced same similar N14 chain, and this red color strand produced a newly synthesized N14 chain. So only two molecules are of intermediate and of light. Remaining totally light chains are there. This pattern could only have been observed if each DNA molecule contains a template strand from the parental DNA. Thus, replication is semi-conservative model. So, in this way, we can prove that DNA replication process done in semi-conservative model only. Next, in the prokaryotes, the replication process having four characteristics. One is it is a semi-conservative replication model. Another one is bidirectional replication. And the third one is semi-discontinuous replication. And fourth one is high accuracy, complexity, and a rapid process. Just now we explained that semi-conservative model means in daughter DNA molecule, one of the old strand, another one is new strand. That is semi-conservative model. Okay. Next, bidirectional replication means at the origin of replication site, the replication fork move bidirectional, that is opposite to the ORI site. Bidirectional process means in this direction synthesis takes place, and opposite direction is also newly DNA synthesized. In the bidirectional process, replication is going on. And semi-discontinuous replication. Semi-discontinuous replication means in one side we can observe one of the parental strand produce new DNA strand continuously. In the opposite parental strand, it produced newly DNA synthesized in small fragments. In this way, we can call this discontinuous. This is called as continuous. And then both type of replications are seen in semi-conservative model. That is continuous and as well as discontinuous. This is called continuous and this is called discontinuous. Discontinuous means the newly synthesized DNA in the form of fragments. It is continuously synthesized. So that is called semi-discontinuous replication. And high accuracy. High accuracy means high fidelity of the DNA. That indicates the information which stored in the DNA molecule is very accuracy by testing or proofreading by different type of DNA polymerase enzymes. That's why it's having high accuracy. And this process involved with various type of proteins and enzymes and energy utilization. So that's why it is complexity. And moreover, the addition of new nucleotides to new strand in a rapid way. That means the total replication process done in with hours only in prokaryotes. So that's why the replication process high accuracy, complexity, and rapid. So what are the basic requirements of replication? Replication is nothing but a duplication of the DNA molecule. It requires some components. So for example, first one is substrates. Substrate means without new nitrogen bases, there is no chance to produce a new DNA molecule. That's why four type of deoxyribonucleosides triphosphates are needed. That is deoxy-GTP, deoxy-ATP, deoxy-CTP, and deoxy-TTP. That is guanine, adenine, cytosine, thymine, which containing with three phosphate groups. They are must to synthesize a new DNA molecule. Second one is basic requirement is template. Template means in the parent DNA molecule, one of the strands should be act as template. Based on the information on the template, the new nucleotides are joined together and synthesize new strand. It is required to direct addition of appropriate complementary nucleotide to the newly synthesized strand. Each strand of parent DNA molecule acts as template for the synthesis of new strand. And the next one is primer. Without primer, there is no initiation of DNA synthesis. DNA synthesis cannot initiate without a primer. Primer is a short piece of RNA molecule. Primer is a short piece of RNA molecule. It is synthesized by RNA polymerase. It is also known as primase. This enzyme helps in the synthesis of RNA primer. Next one is enzymes and proteins. Various enzymes and protein molecules involved in the replication process. We can discuss further here. So, for example, different type of proteins and enzymes, how they are perform the function in the replication process. So, for example, the DNA A protein, which is a molecule which recognizes the ORA site in the DNA molecule. ORA site is nothing but it is a starting point of replication in the DNA molecule, which is recognized by DNA protein. In the DNA molecule, both the strands are helix or spirally coiled. These spirally coiled two strands are separated or unwinding by one of the enzyme, that is helicase. Helicase is necessary to remove the coils or spirals in the DNA molecule. So, and it requires ATP energy for the process. Another one is SSB, single stranded binding proteins. These single stranded binding proteins are stabilize the DNA molecules in a single stranded fashion. They prevent from the double stranded formation. These proteins are prevent the double stranded formation. It is also must in the replication process. Another one is a short piece of RNA molecule synthesized by one of the enzyme, that is primase. It is a RNA dependent DNA polymerase. It is a DNA dependent RNA polymerase. So synthesis of RNA primers also very important. During the elongation process or the extension of the DNA molecule by three enzymes, that is DNA polymerase enzyme, we can discuss separately. and joining the Okazaki fragments by one of the enzyme, that is DNA ligase. It requires NAD. And DNA topoisomerases class 1 and 2 both are required for the removal of positive supercoils ahead of advancing replication forks. After the formation of replication fork, to prevent the supercoiling of DNA molecule by a group of enzymes that is known as topoisomerases. So after helicase enzyme unwinding the DNA molecule, then a immediate problem is resolved by the DNA topoisomerases, which are two types, topoisomerase 1 and topoisomerase 2. So next, the crucial role in the replication process is DNA polymerases. The DNA synthesis is catalyzed by a group of DNA-dependent DNA polymerase. They are commonly called as DNA polymerases. There are three types of polymerases in prokaryotes. That is DNA polymerase 1, polymerase 2, and polymerase 3. In case of eukaryotes, 5 types of DNA polymerase enzymes are there. One is alpha, beta, gamma, delta, and epsilon. In the eukaryotes, the DNA replication process is more complexitive and a time-taking process when compared to prokaryotes. First, we can go through the polymerase 1 enzyme. First time it is discovered by Arthur Korenberg. That's why it's called Korenberg enzyme also. It involved in the removal of primers. The short piece of RNAs removal are excised by the polymerase 1, and the gap filling on lagging strands. The primers replaced by new nucleotides, and another one is DNA proofreading and repair also. These are the functions of polymerase 1. The polymerase 1 structural subunits only one. The molecular weight nearly 1,03,300 Daltons. DNA polymerase 1, 3 to 5 exonuclease activity and 5-3 exonuclease activity. So both are, we can see in this only enzyme. In the remaining polymerase 2 and 3, only one kind of activity, that is 3 to 5 exonuclease activity we can see. But the extraordinary feature of polymerase 1 enzyme is 5-3 exonuclease activity. That means any mutated segment present in the DNA molecule, it will be immediately removed or cut by the action of polymerase enzyme 1. The polymerization rate nearly 16 to 20 per second. Okay? Nearly 16 to 20 nucleotides added to the new synthesized strand based on the template information. So next, polymerization, DNA polymerase 2. DNA polymerase 2 also helps in the DNA proofreading and repair. It has structural subunits nearly 7. The molecular weight 88,000 Daltons, and the feature 3 to 5 exonuclease, and the polymerization rate is 40 per second. The main core enzyme in the DNA replication process performed by polymerase 3. That's why it is a core enzyme or main enzyme in the elongation process. It synthesizes DNA strands on leading and as well as lagging strand. The Okazaki fragment synthesized and as well as the leading strand synthesis, both are performed by the polymerase 3. DNA proofreading also. Structural subunits nearly less than or equal to 10. The molecular weight 7,91,500 Daltons. It shows 3 to 5 exonuclease activity. And the polymerization rate nearly 250 to 1000. When compared to polymerase 1 and 2, the polymerization rate high in DNA polymerase 3. That's why within couple of hours in the prokaryotes, the DNA replication process completed. Per second, 250 new nucleotides added to the synthesized strand. Next, we can explain about what is meant by exonuclease activity. Exonuclease activity means the removal of mismatching pairs or any mutated segment in the DNA molecule. If the 3 to 5 exonuclease activity, excise means cut or removal mismatched nucleotides. For example, here the template strand containing G is paired with A. It is not correct as per base rule. G is always combines with C. That's why the function of 3 to 5 exonuclease activity of polymerase 1, 2, 3 remove the adenine and replace with cytosine. That is called exonuclease activity. Only DNA polymerase 1 having this property, that is 5,3 exonuclease activity. 5,3 exonuclease activity cut primer, that is you can remove the primers and as well as excise any mutated segment present in the DNA molecule, that is removed by 5,3 exonuclease activity in the DNA polymerase 1. Three important stages in the DNA replication is one is initiation, another one is elongation, third one is termination. So first, initiation. Every genomic material, it's having a particular position or a spot which initiate the replication process in the eukaryotes and prokaryotes as well as. So in the case of E. coli, the initiation process occurs at the origin of replication, that is known as ORI site. Generally, we can see here that is ORIC, that region initiate the replication process. The structure of origin of replication is identified or recognized by a protein molecule, that is DNA A protein molecule, which recognize the ORI point in the genomic material, whether it is a circular chromosome or maybe a double-stranded helix structure. So this ORI structure of origin nearly 248 base pair long and 80 rich region. So in this ORI region, we can see some conserved DNA replication, DNA sequences are there, which are almost all similar in all prokaryotes. You can see in 3 repeats of 13 base sequences and 4 repeats of 9 base pair sequences. So these are the 3 repeats of 13 mere sequences. Mere sequence is nothing but it is a oligonucleotide which consisting of few nucleotide series, majorly which containing 80 rich. And another side, the downstream side, 9 mere sequences are there. These are the 5 repeats actually. 5 repeats of 9 mere sequences also 80 rich region. Based on this conserved sequences, DNA protein molecule identify the ORIC region. The next part or next process, the DNA B, that is nothing but helicase enzyme, along with the DNA C are joined in the DNA and DNA complex, open the local 80 rich region and move on the template downstream further to separate enough space. So earlier we can say that is ORI. So this ORI identified by DNA protein molecule. Then after DNA B and DNA C are joined together, and DNA B molecule, that is helicase enzyme, open the double-stranded DNA by breaking or denaturation process. It becomes a replication bubble. It forms a replication bubble. This replication bubble at the point of ORI region, it will be proceed towards both directions. It is a bidirectional replication process. Both sides will be replication proceeded up to the terminus region. So that's why we can see after open up the DNA, double stranded DNA molecule, we can see Y-shaped replication forks are formed both sides. So this is one Y-shaped DNA replication fork, another one replication fork, one more side. It proceeds towards both directions. The replication starts initiated in a single point, but it proceeded in bidirectional manner in the prokaryotes. But in case of eukaryotes, number of replicons are formed. Replicon is nothing but it is a unit of DNA replication. These replicons are initiated at various regions. For example, if it is a eukaryotic DNA, see this one here, the replication process is going on, another site and the third site. So these are the different multiple origin of replication present in the eukaryotes. Replication forks move away from each other. So both sides we here also bidirectionally at various points the replication process is proceeded. So next, after a separation of these two strands, the two single-stranded DNA molecules or strands are stabilized by a group of protein that is called as SSB protein, single-stranded binding proteins. These single-stranded binding proteins prevents the formation of double-stranded DNA. So that's why it will be stabilize the single-stranded DNA molecule. Helicase is unwinding the DNA to remove the spirals by breaking the hydrogen bonds between the two strands. So then after, a group of enzymes uh, required for the relieving the transition or a torsion stress in the DNA strands. Those enzymes are called as topoisomerases. So these topoisomerases are prevent the supercoiling of DNA molecules. The supercoils in DNA replication prevented by a group of enzymes called as topoisomerases. Topoisomerases are two types, type 1 and type 2. Type 1 is positive type, it prevents the positive supercoiling, and type 2 topoisomerases are generally called as gyrases. These gyrases prevent the negative coiling of the negative supercoiling of the DNA molecule. So when the helicase removes the unwinding the double-stranded DNA molecule, a immediate problem is there, that is supercoiling of the DNA molecule. Such type of activity prevented by topoisomerases. Just like a telephone wire in the olden days, we can see the telephone wires string interwoven with the same strands. So the spirals are removed by that one. The next one is formation of primosome. Uh, After stabilization of the single-stranded DNA and the transition relieved by the topoisomerases, then next process is formation of primosome. Primosome is a collection of helicase and primase enzyme. So primase enzyme involved in the RNA primer synthesis. RNA primer synthesis. So in this way, in the initiation taken place in the ORA site, and it will be proceeded to both direction up to the terminus region. So next part is elongation. Elongation is the second step in the DNA replication. It involves addition of four type of nucleotides based on complementary base rule, that is A, T and GC of the template strand. Based on the template strand information, one by one nucleotides, addition of nucleotides, that is DNTPs. DNTPs is nothing but deoxy-CTP, deoxy-GTP, deoxy-ATP, deoxy-TTP. These are the four type of nucleotides are joined with the help of DNA polymerase 3 enzyme. So DNA is synthesized only in the 5 to 3 direction, 5 to 3 direction, and never in the 3 to 5 direction for the complementary strand. On both complementary strands, the newly DNA synthesized in 5-3 direction only. This is very, very important point in the DNA replication. So in 3-5 template strand, the DNA synthesis made in 5-3 direction in a continuous way is called as leading strand. The 3' template of the parent DNA molecule synthesize DNA in 5' direction in a continuous model. So that strand is called as leading strand. And the other template, that is 5' template of the parent DNA molecule, it synthesizes DNA in the form of fragments, in the 5' direction only. Those latter, these fragments are joined together and it makes a full-fledged DNA strand. The small fragments of DNA called as Okazaki fragments after the discovery of R. Okazaki. The new DNA is made by this discontinuous method is called as lagging strand. So one of the parent DNA molecule template strand synthesized DNA in the form of short fragments or short pieces which consisting 50 to 100 nucleotides with the help of number of RNA primers. So the leading and lagging both the strands are synthesized simultaneously by a single dimeric DNA polymerase 3 complex. After DNA synthesis by DNA polymerase 3, DNA polymerase 1 uses its 5' exonuclease activity and remove the primers, then fills the gap with a new 5' exonuclease activity. So this is how the strands are going newly synthesized in both directions. We can see in the next diagram, we can easily understand that. So this is parent DNA duplex. So this is the one of the parent DNA molecule. This is another template. In 3' template, this is the 3' template. This 3' template synthesizes a new DNA strand towards the replication fork, continuously. So that strand is known as leading strand. So leading strand always synthesized in 5' direction towards the replication fork. Another parental DNA strand, that is 5' template. This 5' template DNA molecule synthesize DNA in the form of short fragments. These short DNA fragments are called as Okazaki fragments. So in this way, the Okazaki fragments are also produced in 5' direction only. So later these fragments are sealed and it make a full-fledged strand, daughter DNA strand. So the Okazaki fragments are always synthesized far away from the replication fork. These small fragments are synthesized on the lagging strand towards the away from the replication fork. So next we can see the DNA ligase enzyme which join together the two Okazaki fragments and fill the gaps between the two Okazaki fragments. So this is the DNA ligase enzyme. So DNA ligase seal the gaps between Okazaki fragments with a phosphodiester bond. So between one of the nucleotide having hydroxyl free terminus, another nucleotide containing phosphorus free terminus. In between these components, phosphodiester bond formed and it make a complete strand. The last step is the termination process. Termination is the very important and it stops the DNA replication occur at a specific site that is known as TAR site or TER site. So these are the 23 base pair consensus sequences that provide the binding site for the TUS gene, a 36 kilodalton protein needed for the termination. The termination site is also conserved by a group of nucleotide sequences. The molecular weight 36 kilodalton protein needed for the termination process. Here TUS gene is there. TUS gene is prevent the leakage activity, then stop the replication process. Then after the two DNA strands, newly synthesized DNA strands, full-fledgedly separated and make two DNA daughter DNA molecules. TUS protein binds to TER elements and stop the DNA B from unwinding DNA. So DNA, it will prevent the DNA B activity. That is nothing but leakage activity. So leading strand replicated up to TER elements and the lagging strand replication stopped 50 to 100 base pairs before ter elements. So this red color spot indicates the termination of replication, so which containing TUS gene. So TUS gene is encoded by a protein molecule. That protein binds the ter elements and stop the DNA B activity. So in this way, we can easily understand in the prokaryote, it is a circular form of chromosome. This is ORI region. The, at the at the exactly ORI region, the replication process proceeds in both bidirectional process up to terminus region, So this is up to terminus region, and it made two daughter DNA molecule. So in this way, we can explain the DNA replication process. But when compared to eukaryotes, it is very simple. But in eukaryotes, the number of DNA polymerase are involved, and proteins also um, required in many number, and it very complex process which occur in the nucleus. But in case of prokaryotes, this process completely totally occurs in cytoplasm only, because it is a poorly developed nucleus is present in the prokaryotes. So the nearly 4 to 5 hours you can complete this process by addition of the new nucleotides with the help of group of enzymes and proteins. So that is known as replizome. Replizome is nothing but it is associated with a group of proteins and enzymes which are required for the DNA replication process to produce two daughter DNA molecules. So once again, we can recapitulate this DNA replication process in prokaryotes. Okay. In the next class, we can go for another topic. Thank you, one and all.